It's just a science project.
Silent breed is people!
A doctor friend once said the same thing to me. Frankenstein was his name.
It's alive! It's alive! It's alive!
That sounds like something out of science fiction.
Please explain to me the scientific nature of the whammy.
We live in a spaceship, dear.
So? Yes, science!
Program complete. Enter when ready.
Hello and welcome to Science on Top. This is episode 209 for Sunday the 29th of November 2015. I'm Ed Brown and joining me today is Dr. Shane Joseph.
Hello.
And Penny Dumsday.
Hello.
And coming up a little later on, we'll be talking about the incredibly diverse DNA of tardigrades and making worms temporarily grow heads and brains of other species. But first, Shane, there's bad news in what I would assume Fox News calls the war on bacteria, as evidence from China suggests we could soon see bacteria that are resistant to every known type of antibiotic. Our last resorts are no longer last resorts.
Well, yeah, I mean,
they are last resorts. They're just ineffective in some ways.
Well, they're ineffective. I mean, there are resistant strains to these antibiotics. Now, to be fair, this has actually happened before. In that I've read articles and heard news about things like vancomycin, which for a long time was one of our last antibiotics that we were able to use to treat pretty much everything. The last even things like golden staph, which is a horrible hospital infection that is resistant to pretty much every known kind of antibiotic, except for I think vancomycin. But I think there have been strains that have been detected that can resist that. Now, in this case, this is another one. I've never heard of this antibiotic. They're called polymyxins, and they've been called last resort antibiotics, and they've been used to fight a certain kind of bacteria called. It's broadly classified as a so-called gram-negative bacterium, and this includes things like E. coli, Klebsiella, Pseudomonas. Usually, they're hospital bugs, and they're usually things that can cause food poisoning and/or horrible sort of hemorrhagic fever. Intestinal upsets and things like that.
Lovely stuff.
Yeah, lovely stuff. Now, as I said, in the past we've had the fallback antibiotic of these polymyxins to treat them. Unfortunately, they've detected a few strains in China which are resistant to these antibiotics. Apparently, the resistance to this antibiotic, in the past we found other resistance to it, but it hasn't been easily transferable. Now they've found these Chinese groups have found this resistance gene on a plasmid. Now plasmids are basically little circular bits of DNA that will be transferred very easily from bacteria to bacteria, and therefore it's extremely. This resistance now will hypothetically spread like a wildfire. Because it will be shared across bacterial populations much easier than say had it been chromosomally based. Now, the reason we think that this resistance is popping up is because of the amount of antibiotics farmers are using in piggeries, and unfortunately, it's a widely used practice all over the world that you feed animals antibiotics to essentially put on muscle, and it's in cows. I think it's used to stop bacterial bloating.
Oh, that sounds good.
Yeah,
bacterial bloating.
Well, basically, the story is that we feed in feedlots, especially cows are fed corn-based diets, which causes them horrible bacterial infections, and we pump the cows full of antibiotics, and it stops them from getting these bacterial infections. And the reason you feed them corn is because corn's very cheap, and it's a very useful food source. Then you can put on a lot of muscle very quickly. I'm not really sure why there's basically a correlation between feeding feeding a farm animal a lot of antibiotics and putting on a lot of meat.
Yep.
So that's what they use these antibiotics for. Unfortunately, this then gets washed into the water sources, into the ground, and more bacteria get exposed to these things. And because bacteria are very good at evolving resistance, they do. So, yeah, that's basically what's happened with this.
So we're doomed.
Yes. Well, it depends who you believe. Basically, there was a worry for a while that we had sort of hit the end of our knowledge about antibiotics. So every antibiotic that we discovered from bacteria, and bearing in mind that most antibiotics that we have are produced by bacteria.
Yeah.
And they're basically a bacterial weapon of war. And that is why other bacteria have resistance to them, because there's always an arms race between different bacterial species to try to compete. Now, we've used antibiotics really quite irresponsibly, from you farmers using them as basically growth hormones to doctors overprescribing them. Now, there was a worry for a while that we'd basically, because we couldn't grow any more bacteria or any new bacteria from soil, things like soil, that we had run out of antibiotics and that we weren't going to discover any more. But now, first of all, they've discovered some new classes apparently, using sort of extra techniques basically to grow new bacteria out of the soil. That's a whole other kettle of fish, and I have real problems with that. But I won't get into it because it's just I'll talk all night. It'll be ridiculous.
You're allowed to talk about soil bacteria as much as you like.
Not all night, he isn't. I've got things to do.
And no one cares, let's face it.
Aww.
But the point is that there are now new techniques to basically discover, hopefully, new classes of antibiotics. That, and there's also a lot of work going into sort of synthetic antibiotics, and taking known antibiotics and changing a few things here and there, and that there's a fair bit of promise in that. So I don't think we're doomed. It'd be nice if we could, say if we could sort of, well, basically, first of all, limit the amount of antibiotics that we pump into farm animals. That would be nice. Yeah. Also, it'd be good if we could stop doctors from over-prescribing these things. And to be fair, they are. A lot of doctors now will not just prescribe antibiotics as a sort of a just
like
in case
preventative sort of, yeah.
Yeah, which is what a lot of them did, and some still do, but not as much as they used to. And also, if we can combine this with just a bit more awareness of how antibiotics work and how we can limit antibiotic resistance, and combine it with these new technologies and these new classes coming up, I think we'll probably be okay.
Phew. Well, I'm glad to hear it.
Yes.
So I guess it's like most things, it's not the end of the world, but things have just gotten harder and tougher. And we need to take more steps to slow the evolution of bacteria against antibiotics, and also develop new tools and new weapons. So,
well, one way of doing that is by a lot of people. Well, again, I'm not sure what it's like now, because there's a lot better awareness of it now. But it used to be that a lot of people would have antibiotics and not finish their dose.
Yeah.
They wouldn't finish their prescriptions. Now, at first glance, you think, "Oh, what's the problem with that?" The problem with that is that you could develop, if you don't finish the entire dose, there could be strains in your body that are sort of borderline susceptible to the antibiotic that won't be killed by your half dose, but would have been killed if you'd taken the full dose. They can then go on and develop stronger resistance.
Yep.
And that's one of the main problems. Well, that is a main problem with antibiotic resistance. People not finishing off the script.
And that's one of those things that we as ordinary people can do to combat this resistance things. We can all make that small step when we get antibiotics to take the whole course. But obviously there are much bigger steps that need to be taken from a regulatory and
just to ask the doctor, is this really necessary? Do I really need to take antibiotics? Because as I said, a lot of doctors in the past would prescribe you antibiotics for flu just in case you got a secondary infection. Now, flu's a virus, so antibacterials aren't going to work on the flu. And there's an off chance you might get a secondary lung infection, but until that actually happens, maybe you should just be
better off risking it, perhaps.
Well, if you live in a Western country, probably.
Cool.
Yeah.
All right. Penny, biologists at Tufts University have induced one species of flatworm to grow the heads and brains of another species, or several other species of flatworms, without altering their genetic sequence. And before we go on, I must point out that we weren't looking for another worm story this week. It's just the fourth week in a row that a particularly cool story has come out about tapeworms or hookworms and now flatworms. It's that time of the year, I guess, when the worm stories happen.
The worm stories, it's like the silly season for worms.
So what happened here?
This is really interesting because I think, we're all taught at school that essentially, neo-Darwinian genetics, and it's the chromosomes and your genes that determine everything. And we're now coming to realize that epigenetic factors or other factors can control development. And what I thought was really interesting, because flatworms are quite known for being able to regenerate themselves, and if you split them in half in the right way, they can grow another head. But they're very flexible creatures. So at first I thought, oh yeah, they've somehow swapped out their heads. But they did this without altering the worm's genomic sequence. So there's no genetic modification here. There's not even a lot of physical modification. What they did is they manipulated gap junctions. And gap junctions are places where cells can communicate with each other. So a flatworm's body is made up of lots and lots and lots of little cells next to each other. And we think of these cells as discrete little bags of stuff, but there's actually a lot of communication, and electrical signals and actual molecules can pass in between cells. And what they did is, by modulating the way that these cells were connected, they were actually able to get one species of flatworm, Giardia dorotocephala, and
nailed it.
Yep, thank you. And they got this flatworm to essentially grow the head of 3 other different species of worms, and this is without modifying its genes, but just by affecting the ways that the cells can communicate with each other. So what's really interesting about this is it's really giving us a new idea about how our physiological function can determine body shape. So when you think about birth defects or healing after injury, that's really interesting, is how can we get a body to grow in a way that's not just its quote-unquote sort of normal developmental process? How can we get it to do something different?
Yeah, I guess it's the sort of thing that the more we learn about it, the more we could maybe use it. Obviously, I think we're a long way from humans. But,
there's a long way from flatworm head to a new ear.
I'm not suggesting that we start swapping the heads of people with chimpanzees or something. But in terms of curing, although that's... Shane's got that evil maniacal laugh there.
I guess the other thing too that was quite interesting is these are different species. The more closely related the species was, the better this worked. So I don't think we're going to be able to somehow recreate a human ancestor or any,
But it's a slippery slope, Penny.
Oh, Shane.
Well, I was more thinking about we can fix genetic defects or something like that, perhaps. But the other thing is it wasn't a permanent change.
No. Well, the flatworms are actually really known for regenerating. And a couple weeks after they sort of
had their new head.
Had their heads changed. Yeah, had their new head. How do you say that? They actually started to regenerate back to their original. So this wasn't a permanent thing. But I still think it's very interesting in that it was not a genetic thing, and it's not even the usual kind of thing we think of as epigenetics, which is methylation or, little sort of sticky notes stuck on DNA to say, do this, don't do that. So this is
this is an entirely external sort of factor. It's not touching the genome at all. It kind of makes me think of Odo in Deep Space Nine, and someone's sending some signal that makes him warp into a different shape all the time, which would be bizarre, but that's the way my mind works.
And kind of cool.
And he could rest in his little bucket.
He's pale, yes.
Yeah.
Yes.
What's the difference between a pail and a bucket?
One's American, one's not.
Oh, is that all it is?
I guess. I don't know. I'm just kidding.
No, that's really cool. It'd be interesting to see if that can be extended to other animals, or if it's purely because of that flexibility that flatworms have. But very cool, one to follow. And Shane, researchers from the University of North Carolina at Chapel Hill have sequenced the genome of the nearly indestructible tardigrade. It's all the microorganisms today. And they've found that nearly 1/6 of the genome is from foreign DNA, as in it's just grabbed DNA from from bacteria or other organisms and incorporated that into its own makeup.
Yeah, which is weird because, well, first of all, I think it's the most foreign DNA they found in a multicellular organism.
Mm-hmm.
I think the last one, I can't remember what the previous record was, but it was about half that, essentially. So these little water bears, we've talked about these little guys before. They're basically indestructible. These things can survive the vacuum of space. It's kind of scary. They're about a millimeter across. They're little, tiny little swimming things that you'd barely take. You wouldn't even know they were there. And yet they can, they've come under a bit of scrutiny recently just because of the fact that they are so good at surviving. We talk about extremophiles a lot, and that usually covers bacteria or archaea, which is like another single-celled sort of something similar to bacteria on the face of it, but not really genetically. But anyway, and we're used to thinking about those things in terms of extreme environments, but tardigrades can survive desiccation, as I said, vacuum of space, extreme heat. And one of the reasons I think they do this is they basically form these little sort of essentially like resting spores. They sort of desiccate themselves and go into like an animated suspension, essentially. Now, the reason they think, or one of the ways they think they're acquiring all this foreign DNA is that when they start to sort of come back to life, their membranes are a bit leaky, and so anything that's around them in their environment just passes through, like DNA-wise, into their cell nucleus, and it will just get incorporated into their DNA. Now, obviously, some of those will be lethal and will just kill some of the tardigrades, but some of them will be incorporated and will actually become more useful. And that's one of the reasons I think that they're postulating anyway that tardigrades are so good at surviving, because they're picking up new genes very, very, very quickly, and anything that is successful will continue to be successful and propagate. So it's horizontal gene transfer, which we know happens in bacteria, that you get genes that will swap between cells without any like progeny. Like we're used to thinking of DNA transfer in terms of mother to daughter cells. But this isn't the case with horizontal gene transfer. It just basically gets swapped. And as I said, we know this happens in prokaryotic life, like bacteria, but we're not used to thinking about this happening with multicellular organisms, but it obviously happens here. So, yeah.
Very cool. But presumably it doesn't just get DNA from other species. It has to somehow sort of presumably get rid of the ones that are, to use a different phrase, junk DNA, things that don't actually help it.
Well, no, I think what might just happen is if it gets integrated randomly, if that's not useful, it'll either just stay there and do nothing, or the
tardigrade will die.
Will die,
and it won't pass it on.
Which is how evolution works anyway.
Yeah,
if you have a lethal mutation, the line goes. But if that mutation is beneficial, it will get passed on. And this is probably what's happening here, just at a sort of a different. It's not Darwinian. Well, it is Darwinian, but it's not, as I said, passed down as we think about it. It's basically you grab a bit of DNA just randomly, and if it doesn't work, it doesn't work. But yeah.
Yeah, but it just, and I know I'm going to sound a little like a creationist, but it sounds amazingly convenient, if you will. Like the tardigrade has just developed all these survival things. Obviously, it's been around billions of years, I would imagine, but it's kind of developed this really sophisticated, convenient armory of defenses.
Yeah, but
then if it hadn't, it wouldn't have been here.
Yeah, exactly.
Yeah.
And B, you don't see the failures because it dies.
Yeah.
What we're seeing is the results of the success.
Yeah.
And that's very, it's a bit sort of, it's, as you said, very convenient.
I find that really freaky when I think about, that aspect of evolution is that,
how many species have come before that didn't make it because of something? And you look
at everything and you go, oh my God, that thing is like really great at living in its particular environment.
And what's even creepier is that we'll think about all the things that were very transient and didn't get preserved in the fossil record because there weren't enough of them.
Yeah.
So we're looking at the past, and I reckon we're missing a heap of stuff just because stuff wasn't preserved because it wasn't there for long enough to be preserved.
And I think also the best way to think of evolution with things like this is just to remember that we're talking 4 billion years. We sort of think about, and a gene here and a gene there, and suddenly you've got this huge armory of genes and defenses and that. But it's so slow that it happens over so many billion generations of these things. But,
it is slow. But then a mechanism like this will speed it up.
And if you evolve something like this, of course you're going to keep it.
Yeah.
Of course, yeah.
But the thing that I also find is just weird is it's from other species. It's a bit like the tapeworm thing where it's growing different species, but not even, I imagine, it's not even species. This is other organisms completely different.
Other kingdoms of life.
Yeah.
Yeah.
That's very cool.
And look, a lot of that will probably just sit there and do nothing. Because it won't integrate into they've sequenced this tardigrade's genome, but the thing is, I wonder how many different species of tardigrade they've sequenced.
I didn't even know there were different species of tardigrades.
I'm guessing there probably would be.
I don't think there's a huge number, but I might be wrong.
Strange. There'd be, yeah, I mean, I'm guessing there'd be different strains. There'd be different, in different areas, there'd be different kinds of tardigrades. I don't know enough about tardigrade taxonomy to hazard a guess, really. But I would guess, considering they're so good at sort of incorporating foreign DNA into their genomes, I'm going to guess that there is a lot of different types. Just educated guess. And I do wonder just how different they are between the different types. And if you did another sequence, say, because sequencing genomes is now pretty easy. I'm not sure how big the tardigrade genome is, but I don't imagine that it would be an arduous task to do another few sequences of different kinds of tardigrades and just see, does this hold for every single? Is this across the spectrum? Like, do you see this everywhere? Do you see the same foreign DNA or the same sort of ratios of foreign DNA in every single type?
Yeah, it's, as you say, it's horizontal gene transfer. We see it all the time in bacteria, but just the crossing of organisms thing blows me away.
Yeah.
That's very cool. Okay, well, that's our show. As usual, all the links are on the web at scienceontop.com/209. And we'd love to hear from you, of course, so please leave us a comment there, or you can drop us an email, feedback@scienceontop.com. And thank you for joining me, Shane and Penny.
No problem, Ed.
Always a pleasure.
And thank you, everyone, for listening. We'll be back again next week, putting science on top of the agenda. Join us then.
You probably never notice them because they're so small, about the size of a pinpoint. But they're tough. The tardigrades have survived all 5 mass extinctions. They've been in business for 500 million years. We used to think that life was finicky, that it would only take hold where it was not too hot, not too cold, not too dark, or salty, or acidic, or radioactive. And whatever you do, don't forget to add water. We were wrong. As the hardy tardigrade demonstrates, life can endure conditions that would mean certain death for us humans.